This video tutorial is all about genetic modification. Genetic engineering is changing the traits of one organism by inserting genetic material from another organism. The organism receiving the genetic material is said to be genetically modified. Now, you need to understand how restriction enzymes are used to cut DNA at specific sites and how ligase enzymes are used to join pieces of DNA together. So first of all, the gene that is to be isolated needs to be located in the original organism. So for example, this could be the gene for human insulin. Restriction enzymes are used to isolate the required gene, leaving it with sticky ends. So restriction enzymes are enzymes that cleave DNA at specific sites to obtain desired genes and leave what we call sticky ends, which are short sections of unpaired bases. A vector, which is usually a bacterial plasmid or a virus, needs to be cut with exactly the same restriction enzyme, leaving it with corresponding sticky ends. So in order to incorporate the gene of interest into the vector, the same restriction enzyme must be used. That's so that the sticky ends of the gene and the vector will overlap via complementary base pairing, so that they can then be joined together by ligase enzyme. This also helps ensure the gene is inserted in the correct orientation. Now, a ligase enzyme is an enzyme that forms bonds between a vector and the gene that's going to be joined with it. If two pieces of DNA have matching sticky ends because they've been cut with the same restriction enzyme, the ligase will link them to form a single unbroken molecule of DNA. The vector and the isolated gene will be joined by ligase enzyme to form what we refer to as a recombinant plasmid that can then be incorporated into an appropriate host or organism to express the desired trait encoded by the gene of interest. That term I just used, recombinant DNA, that's DNA that is an artificially formed section of DNA as a result of the transfer of genetic material from one species to another. So you also need to understand how plasmids and viruses can act as vectors, which take up pieces of DNA and then insert these recombinant DNA into other cells. So the term vector just means a vehicle that is used to deliver recombinant DNA into a target cell. A vector artificially carries foreign genetic material into another cell where it can be replicated or expressed. The two main types of vectors are bacterial plasmids and viruses. So bacterial plasmids are just circles of DNA which can be found in bacteria. And then when the bacteria reproduces, the plasmids are copied as well. So the recombinant plasmid can quickly be spread as the bacteria reproduces via binary fission and will express the gene. Viruses will carry recombinant DNA and infect organisms that they come into contact with, making the host organism express the recombinant DNA. Now, you need to be aware of the case of diabetes and specifically the need of diabetics for insulin and how large amounts of human insulin can be manufactured by genetic modification in bacteria and how they're then grown in fermenters. So individuals with diabetes are unable to produce enough insulin. As a result, insulin needs to be injected to help lower blood glucose levels, converting it from glucose into glycogen, which is insoluble and so can be stored in the muscles and the liver. Insulin must therefore be produced in large quantities to help treat diabetes. Now, prior to the development of genetic engineering, insulin for humans was collected from pigs. Now we use genetic engineering. So bacteria are genetically engineered to produce human insulin. which can then be collected and purified for use with absolutely no fear of rejection, which was one of the issues of using pigs, as well as sort of issues relating to ethics. Once some bacteria have been genetically engineered, they're grown in large quantities in a fermenter. Each bacterial cell will produce a tiny mass of insulin, but using a fermenter, large quantities of insulin protein can be produced, and then they're extracted and purified for use by diabetics. So actually, how does this happen? Now, we've gone through this already, so it's the same process as we looked at before. We've got a restriction enzyme, which is used to cut out the insulin gene from the human DNA. The restriction enzyme will also be used, so the same one, to cut open the plasmid DNA of a bacteria. The insulin gene will then be inserted into the plasmid DNA of the bacteria and joined using ligase enzyme. The insulin gene is now recombinant and will be inserted into the bacterial cell, which is its vector. And the transgenic bacteria is now complete. So it contains the genes from different species and will be grown in a fermenter to produce again and again and again, duplicating by binary fission. In addition to understanding how we reproduce insulin using genetically modified bacteria, you also need to understand how genetically modified plants can be used to improve food production. So genetically modified plants are plants that have foreign DNA inserted into their genome. This is usually done to improve the food production in some way. So, for example, tomatoes can be genetically modified to make them grow larger. Wild rice can be genetically modified to produce beta-carotene, which is needed by humans to make vitamin A. Crop plants can be genetically modified to be resistant to herbicides. So the list goes on and on. Now, again, the process is the same as we've seen before. The same restriction enzyme is used to cut out the gene for pest-killing protein from the bacterial plasmid, and the same restriction enzymes are used to cut open the plant DNA. Now, these CHENs that are produced will enable an overlap in the bases that are exposed, which can then be joined together by ligase enzymes. And those overlapping bases and cutting it with the same restriction enzyme will ensure that the gene's inserted in the correct orientation. This will form a recombinant DNA, which can then be inserted into a plant cell, and then the transgenic plant is grown to express the desired trait. So in this case, it could be pest resistance. Now, there are a wide range of different advantages and disadvantages of genetically modified crops. So obviously, genetically modified crops are going to have beneficial characteristics. So they might have greater vitamins or nutrients in them. They might have increased chances of survival against things like pests and disease. They might have a greater crop yield, which is obviously going to yield more sort of financial benefits for the farmers. And it might be that they can grow in multiple different seasons. So your kind of growing season can be extended, which also will enable a more stable food supply. However, it's not scientifically proven to be entirely safe for human consumption. We still don't really know what its impacts are for us. It's leading to a loss of biodiversity because fewer weeds are able to survive. Seeds for genetically modified crops can be really expensive. And also there's a risk that genetically modified crops could crossbreed with wild crops and contaminate them with entirely unknown, unexpected results. Now, finally, you need to understand the term transgenic. Transgenic means the transfer of genetic material from one species to a different species. This means that a transgenic organism contains genes from a different species. So, for example, the recombinant bacteria used to produce human insulin are transgenic as they contain genetic material from a human. Over the course of this tutorial, we have quickly sped our way through the specification points listed on the slide in front of you. Have a little read through and see if you are confident with each one.